Antibodies are very specific. They combine a particular protein among tens of thousands. They have two arms, identical, that simultaneously bind to the same protein. Bispecific antibodies are engineered molecules that contain arms from two different antibodies. So they combine simultaneously two different proteins. T-cell bispecifics typically contain one arm that binds to the T-cell receptor and the other arm that binds to the tumor-specific antigen or tumor-associated antigen. So they actually guide lymphocytes against tumor cells and mediate the destruction of the latter. HER2 is a proto-oncogene that can be activated by mutation or overexpression. There are several lines of therapy against tumors in which HER2 is activated. If it is activated by overexpression, there are therapies based on monoclonal antibodies and on small molecule inhibitors that attenuate signaling. And against tumors with mutated HER2, the second therapy can also be used. This is chemical inhibitors of the signal transduction of HER2.
So we observed killing of the tumor cells. What we do in the lab is that we co-culture tumor cells from patients with lymphocytes from the very same patient. And then we add the TCB. And in just 2 days, we observe a specific killing. And we also perform experiments in mice, and we saw the very same effect. But what is most remarkable about our TCB is its specificity, that it only kills tumor cells. And we say that because we've done several experiments using normal cells, and there's no effect on these type of cells because normal, healthy cells do not express P95HER2. So nowadays, metastatic HER2 positive breast cancer tumors are being resistant to the current treatment. So there is a need for a new type of treatments for these patients. And immunotherapy is being a very promising therapeutic approach, provided that the T cells are directed to the tumor site. And that's precisely what our TCB does.
Since we have found that P95HER2 is exclusively expressed in tumor cells and is not expressed whatsoever in healthy tissue, we can use it to deliver additional therapies against cancer cells. These may include chemotherapeutic agents bound to the antibody, or may include CAR T cells specific for the tumor cells expressing P95HER2.